me go back. Okay, great. Yeah, so today I'm going to talk about traumatic stress and accelerated cellular aging, which is work that my colleagues and I have been doing for the last, I don't know, 7 to 10 years or so, which has led us down a particular path. And I'll try to give you the bird's eye view of this body of work and what we've found. So what do I mean when I'm talking about accelerated aging? Well, we know that there are numerous biomarkers and biological systems that change with aging. And this includes epigenetics. And I'll be talking a lot today about DNA methylation and a particular algorithm that is derived from DNA methylation data referred to as DNA methylation age, although certainly that's not the only epigenetic phenomena that changes with age. We know metabolics change with age. We know inflammation increases over the lifespan. In fact, there's a construct called inflammaging, which refers to sort of chronic low levels of inflammation that tend to increase over time. With age, there are changes in HPA axis and other stress response systems. And for some people, these changes can occur at a pace that is faster than what might be expected based on chronological age. So for these individuals, we think of their biological or cellular age as exceeding their chronological age. And we got onto the story about how traumatic stress might be associated with accelerated aging by noting some of the associations between traumatic stress and a variety of sort of cardiometabolic and neurocognitive health conditions that seem to be more prevalent among individuals with traumatic stress. But what do I mean by traumatic stress? So people define this differently. Some people are talking about sort of trauma exposure. That's generally not what our lab is focused on, because actually trauma exposure, even repeated trauma exposure, is incredibly common in the population. With by adulthood, about 90% of individuals have been exposed to a traumatic experience. But we are often more focused on the psychiatric sequelae of trauma, including post-traumatic stress disorder, which we define by 4 symptom clusters in the DSM. So those include re-experiencing of traumatic events. So this is reliving of the event itself, flashbacks, nightmares, intrusive imagery, and so on. Avoidance of trauma-related memories, feelings, and events that prompt those types of internal cognitive and emotional experiences. Emotional numbing and negative cognitions about the self, the world, other people. And then symptoms of hyperarousal, so including things like startle, hypervigilance, anger, poor concentration, sleep disturbance. And these symptoms of PTSD tend not to occur in a vacuum. So following a trauma, it's not just PTSD that can occur, but we know that there's a variety of different forms of PTSD comorbidities that are actually the norm, not the exception to the rule. And these really span the internalizing and externalizing dimensions of psychopathology. So internalizing dimension includes things like depression and anxiety. So we see a lot of comorbid generalized anxiety disorder and panic disorder with individuals with PTSD. And then on the externalizing side of things, we think about comorbid substance use disorders and even antisocial personality disorder. And we've shown before that PTSD is strongly correlated with both internalizing and externalizing dimensions. And it's likely that variability in temperament and personality influence whether someone's going to have a kind of PTSD plus kind of comorbid internalizing presentation or a comorbid externalizing presentation. So we really think it's those psychiatric symptoms that are chronic traumatic stressors that serve as both emotional and physiological pathways that increase health disparities in the form of accelerated cellular aging. And so we're focused on that story primarily, although we always look at whether it could be the trauma itself and not the psychiatric response to the trauma. So this is a conceptual model that my former postdoc, Philemon Morrison, from a few years ago, and I worked on together, where we talked about at the top there, you see chronic PTSD symptoms, although really that could be, chronic traumatic stress. So including psychiatric comorbidity, interacting with genotypes. So both direct effects and moderated effects by genotypes that might put one at risk for accelerated aging, leading to biomarkers of advanced cellular aging. So this would be in DNA methylation and gene expression, which I'll talk about today. It would be in inflammatory and immunosenescent profiles, and in things like telomere length shortening, although we've done some work on that, I won't highlight that today. And then those biomarkers of accelerated cellular aging, they tend to give rise then to the clinical manifestations of accelerated aging, which is how we got onto this whole story. So this would be like early onset of neurocognitive decline, including things like Alzheimer's disease, early onset of metabolic and cardiovascular diseases, and even early death. And on the left side of that figure, you see that red arrow that says chronological aging. And that's really just to kind of remind people that all of these pathways that we are hypothesizing are occurring in the context of chronological age. And we do expect that there may be critical windows of development when the kind of iatrogenic effects of this environmental factor, traumatic stress, and the at-risk genotypes may be greater than at other developmental stages. We also like to think about the fact that for individuals who are younger, if they have chronic psychiatric symptoms and at-risk genotypes, they may have more time with these psychological stressors, which serve as, kind of prompting the autonomic arousal and stress reactivity that may then have a kind of more cumulative burden and increase the risk of accelerated aging over time, because there's just simply more years of coping with these psychiatric symptoms if they are chronic. So this is a little figure that just gets at how we got onto this story. And it was our observation about a relationship between PTSD symptom severity and metabolic syndrome over time. And this was in a cohort of young veterans. So they were all Iraq and Afghanistan returning veterans, about 1,500 individuals in this cohort nationwide. And we had assessed them at 2 time points. And what we found was not only was metabolic syndrome more prevalent among those with PTSD, but this is what's called a cross-legged panel model. So we look at the relationship between metabolic syndrome at time 1 and time 2, and between PTSD at time 1 and time 2. And then we look at these cross lags. So we look at how does PTSD symptoms at time 1 predict metabolic syndrome at time 2, controlling for baseline levels of metabolic syndrome. And likewise, how do metabolic syndrome at time 1, how does that predict PTSD severity at time 2, controlling for baseline levels of PTSD? So we're looking at bidirectional associations. And what we saw in these very young veterans who were assessed about 2 years apart was that PTSD symptom severity at time 1 was predicting increases in metabolic syndrome criteria being met at time 2, even accounting for the baseline levels of metabolic syndrome. So this is and the reverse was not true. The metabolic syndrome was not predicting increases in PTSD symptom severity over time. So this was a moment where we said, there's something going on here. What could that be? Related to that, in a distinct sample of returning veterans, so these are all also veterans from Iraq and Afghanistan who participated in the TRACS cohort, which I'll talk a lot about today. We looked at PTSD, metabolic syndrome, and cortical thickness in the brain. And what we found, again, was a very strong relationship between having PTSD and increased prevalence of metabolic syndrome. But also we showed that the severity of the metabolic syndrome was very strongly related to decreases in cortical thickness, really across the brain, although it was primarily or strongest, I should say, in temporal, parietal, and frontal lobes, but there was occipital lobe involvement as well. And we tried to kill this effect. So this is decreased cortical thickness. We tried to kill it by throwing in a ton of covariates that we thought might explain it, by looking at if there were outliers. We did all the things. We kicked the tires on this, and I could not kill this effect no matter what I did. So really appears to be a very strong relationship between increases in metabolic syndrome severity and decreases in cortical thickness. So that, again, got us wondering what could be accounting for this. And so that's how we got onto this story about DNA methylation age. So for those of you who don't know, DNA methylation is a chemical change that occurs at specific DNA sequences throughout the genome. So, unlike your DNA, which doesn't change except in the context of cancer cells or things like that, DNA methylation are little chemical tags that get added to particular loci in the epigenome, and they impact the extent to which a gene can be transcribed. So whether it can be expressed or not. These changes are definitely sensitive to the environment and also have known kind of alterations that occur with aging. So in 2013, 2 independent investigators I've listed on this slide, Hannum and colleagues and Steve Horvath identified this relationship between DNA methylation and chronological age by identifying loci in the DNA methylation, in the DNA across the epigenome that were really strongly correlated with chronological age. And they each created independent algorithms that would weight the DNA methylation levels across the epigenome by their relationship with chronological age, so that you could sum all this up into one single score that could predict someone's chronological age. And the correlation with chronological age was really remarkable. So a correlation of 0.96 for both of these algorithms, which were developed separately from each other and in different types of tissues. The Hannum metric was developed in whole blood. The Horvath metric is a multi-tissue predictor of chronological age, including like 54 different types of tissues, brain cells, blood, you name it, is all in there. More recently, a team led by Liu et al. Developed this slightly different metric of accelerated aging or DNA methylation age called GRIM age, which differs from the Horvath and Hannum metrics and actually about 17 other DNA methylation age algorithms that are out there. These differ because these were not trained on chronological age, but rather on the ability to predict time till death and physical health. So that could be particularly novel and useful for us in our study of accelerated aging. So today, as I mentioned, I'm going to give you this kind of bird's eye view of our results in this area. And I'm going to talk about studies that have sort of some common methods throughout them and come from one of three cohorts primarily. So we do a lot of work with the TRACS cohort, which is based here in VA Boston, although also has a site in Texas. And this is a cohort of veterans from the Iraq and Afghanistan wars. So at baseline, they're in their early 30s, and they're now up to a third time point of return assessments. And it's a really remarkable cohort. I think the sample size that I have on this slide is actually a little outdated. They're up to about 700 veterans in the cohort now. It's remarkable because of the deep phenotyping they have in this cohort. So we have psychiatric symptoms, we have neurocognitive measures, we have blood draws on them, we have MRIs on them. So I'll be talking about all these different elements when I talk about results we've seen. Our group has been the one to do all the genetic work in that cohort. Then we have a cohort that we collected that are mixed era veterans and their trauma exposed partners. So at baseline, they were in their mid 50s, and we have a 15-year follow-up study going on right now. I have an R01 to bring these individuals back in, get MRIs on them, which will actually be a and again, we've been studying them. We have neurocognitive measures, we have psychological measures, we have a blood draw at each time point, and now we're getting MRI back on them. And then we have a VA National PTSD Brain Bank that we do work with, where there's about 115 brains. We have left hemisphere and 3 different brain regions where we've, again, got DNA methylation from multiple brain regions. We have DNA, we have gene expression. So we're really able to run our analyses in different cohorts, look for replication, look for whether effects we see in blood generalized to blood and to brain rather, and so on. Across these cohorts, for the most part, we have genotyping on the Illumina 2.5 million array. And from that 2.5 million array, we can impute the rest of the genome. We have DNA methylation data using the Illumina EPIC arrays, which give us 850K DNA methylation loci. We have RNA sequencing on most of these cohorts, including those that are at multiple time points. We have metabolic parameters on them. And then more recently, we acquired inflammatory and neuropathology biomarkers using Simoa technology on the living cohorts, the returning veterans and the mixed era veterans. And the Simoa data are really unique because these technologies can assess very low levels of these analytes that we're interested in. So before someone might be showing sort of outward signs of neurocognitive decline, we can detect those early levels of it in the blood. And then on most of these cohorts, we have MRI, both structural and functional brain MRI. And then on my cohort that we're bringing back in, those mixed era veterans, we have neurometabolites through magnetic resonance spectroscopy. So I like to show people this slide. This was the first study that we did focused on DNA methylation age and PTSD. But before I talk about the kind of PTSD effect, I just want to show you the relationship between chronological age on the x-axis here and estimated DNA methylation age. It actually looks the same whether you use that Horvath metric or the Hannum metric of cellular aging, because what's remarkable to me and remains remarkable despite all these years later is that we can take these algorithms that were developed in completely independent samples, different tissue types, apply them to our data, and get this really remarkable correlation of R equal 0.88 between chronological age and methylation age. But what's also notable here is that not everyone is right on this regression line, right? Some people have overestimated methylation age relative to their chronological age. Some people have underestimated methylation age compared to their chronological age. And so we conceptualize these individuals as having accelerated and decelerated cellular aging, respectively. And you can see here in this box on the right, there are actually substantial variability in these metrics. So we've got people who are overestimated by as much as almost 19 years and underestimated by as much as 11 years. And so what we do when we want to model accelerated aging or decelerated aging, it's a spectrum, it's one variable, rather than modeling actual chronological age, is we run a regression in which we take chronological age, we use it as a predictor of methylation age, and then we save the residuals from that analysis. So if you have a positive residual, you're in that overestimated range, or you have accelerated methylation age relative to your chronological age. If you have a negative residual, you've got decelerated or slowed cellular aging compared to chronological age. And that's our primary variable that we work with throughout the rest of the analyses and results I'm going to show you. So this was our first paper where we looked at relationships between neural integrity and PTSD and accelerated cellular aging. And what we did here, because we think that one sort of measure of PTSD symptom severity at any one point in time might not be that predictive of something like cellular aging, we were able to measure using latent variable modeling, sort of the severity of someone's PTSD symptoms from across the lifespan. So we looked at, pre-military childhood trauma-related PTSD symptoms. We looked at immediate post-deployment PTSD symptoms, and we looked at PTSD symptoms within the past 30 days. And this was all conducted in that TRACS cohort. So this is the young sample of veterans who are in their average age in their mid-30s at this, or even early 30s at this time point. And lo and behold, we did find a small but statistically significant association between PTSD symptom severity and accelerated methylation age. And this is controlling for things like ancestry and biological sex and cell types that the DNA were obtained from. So then we wondered, is it really all of PTSD symptoms that are predicting these changes, or might it be specific to certain symptom clusters that we think are more related to kind of hyperarousal and physiological symptoms, and autonomic arousal, elevated heart rate, things like that. So we looked at in this independent cohort, this is our sample that's in their mid-50s, is it the re-experiencing? Is it the avoidance and kind of numbing symptoms, or is it the hyperarousal symptoms and PTSD? So we replicated the PTSD association with accelerated methylation age, but we found that it was actually being driven by this hyperarousal symptom cluster. So again, this is poor sleep, anger, physiological arousal, concentration problems, et cetera, driving the relationship between PTSD and accelerated methylation age. Then we thought, okay, well, those are just our cohorts. Can we see this anywhere else? So we do a lot of work with the Psychiatric Genomics Consortium. I contribute to the PTSD and Health group. It's a subgroup. And I also contribute to the PTSD epigenetics group. So we were able to send our scripts out to everyone else who had relevant data. So they had trauma, exposure, PTSD, and DNA methylation, and get back results that we could then meta-analyze. And indeed, we found a replication at the meta-analytic level. And I should note, this is not just VA data. These are not just veterans. This includes active duty military samples, but also neighborhood trauma samples like the Grady Trauma Project and the Detroit Neighborhood Health Study. So we found here, again, association between PTSD symptom severity and accelerated methylation age at the meta-analytic level. And then I had mentioned earlier in my talk about, the first two methylation age algorithms were developed to really predict chronological age, Horvath and Hannum cellular age metrics. But more recently, there's been this Liu et al. Method called Grim age, which is really trying to predict time till death. So if you factor out age from that, you could look at the extent to which someone's death is sort of more likely to occur more quickly than you would expect based on their chronological age. So my recent postdoc, Sage Han, who's just left our program and gone on to a position at Old Dominion University, ran these analyses. This paper is in press now. where she looked at how not just PTSD, but PTSD and also internalizing and externalizing psychiatric comorbidity was associated with accelerated aging using this metric of shortened time till death. And we did this in two different cohorts, the TRACS cohort, those young veterans, and the middle-aged veterans, which is the cohort that we collected. And we again found in the TRACS cohort that PTSD was again associated with shortened time till death. That didn't replicate in the older cohort, but in both cohorts, we found that externalizing psychiatric symptoms were also associated with shortened time till death. And then, we've wondered about this because all of the data that I've shared with you so far, all those results are cross-sectional in nature. So we've have some opportunities more recently to look at, okay, well, is it really a longitudinal? Is there a directional association going on where the psychiatric symptoms are predicting changes in accelerated aging over time? So in that TRACS cohort, using the first 2 time points of data, we were able to look at this. And again, we did find that certain PTSD symptom clusters were associated with an increasing rate of accelerated aging over time. So we had accelerated aging measured at 2 time points. In this case, it was the PTSD avoidance and numbing symptoms as opposed to the hypoarousal symptoms that was associated with accelerated aging over time, but also the substance use, in particular alcohol abuse and dependence on the right side here of this figure, were associated with an accelerated pace of epigenetic aging. And how you can interpret this is that for individuals who had an alcohol abuse or dependence diagnosis at baseline, they were aging in their epigenome at about 1.5 years for every 1 year in between our 2 study assessments, which were about 2 to 3 years apart. Whereas those who didn't have an alcohol abuse or dependence diagnosis at baseline, their methylation age increased about 0.6 years or 0.7 years for every intervening year between the 2 study assessments. So really helping to get at that directional question. I should note that we actually have replication of that association in another data set of mine that I'm writing up right now, and in a new meta-analysis that one of our students is working on now using data again from the PGC, the Psychiatric Genomics Consortium. So, at that point, having seen this relationship across a number of different psychiatric conditions, different types of traumatic stress, across a number of different data sets, we started to wonder, okay, great. So this relationship seems to be there. Does it mean anything? Is it clinically useful? And so we started to look at what are the health correlates of having advanced DNA methylation age. And what we've seen, this is from a couple of different papers, but I kind of put them all into one slide, is that indeed this seems to be meaningful. So individuals who have accelerated methylation age, they're showing worse performance on tests of executive functioning, on tests of verbal and working memory. They also show some alterations in brain morphology, including reduced microstructural integrity in the genu of the corpus callosum, which connects the right and left prefrontal hemispheres. So is sort of tracking with that loss of executive functioning, given the role of prefrontal hemispheres in executive functioning. We also see decreased cortical thickness in right prefrontal cortex and in the left fusiform gyrus, which is actually critical for things like word finding and naming. And then we show alterations in the blood-based biomarkers. So this includes increases in inflammation like C-reactive protein and interleukin-6 and TNF alpha, increases in metabolic syndrome parameters, blood pressure, blood sugars, things like cholesterol and triglycerides, and then markers of neuropathology, such as GFAP, which is a marker of astrocytic damage, and increased oxidative stress, like in GGT. And some of those blood-based biomarkers are those Simoa markers, which can detect, again, very low levels of the analytes in the blood. So really picking up on early signs of biological changes that seem to be correlated with advanced methylation age. And then in one of our studies, we were actually able to look at, again, the question of directionality. So is it accelerated methylation age that's predicting increases in metabolic syndrome over time, or is it metabolic syndrome at baseline that's predicting increases in accelerated aging over time? So this is, again, in that young cohort of TRACS veterans from Iraq and Afghanistan, where we showed actually the direction was really going in just one way. So individuals who had accelerated methylation age at baseline were showing increases in metabolic syndrome over time, even controlling for their baseline levels of metabolic syndrome. And this, when we drilled down into the various components of metabolic syndrome, we found it was really driven by both increases in obesity, central obesity, and dysregulated lipids, so cholesterol and triglycerides primarily. The reverse direction of association wasn't there. I'll say I have replication of this also in a cohort, in a paper that I'm working up now. So if you remember back to that kind of schematic I gave you early on, saying we think that these environmental factors like psychiatric stress or traumatic stress are increasing the risk for accelerated aging, but that it also is likely moderated by genotypes. So we started looking at a particular gene called the Klotho gene that we thought maybe was related to accelerated aging. Certainly by no means is not the only gene we're interested in or the only one that we've looked at, but it's one that has a kind of interesting story because Klotho was very strongly associated with Alzheimer's, diseases of aging, animals who have certain knockout of certain genotypes within the Klotho gene show an accelerated aging profile. So we got onto this Klotho story. And what we did was to look at every genetic variant that we had sufficient power to look at in this gene. And look at how it moderated the relationship between PTSD and accelerated methylation age. I'll note this figure on the right was drawn for us. The story of Klotho is kind of an interesting one. So Klotho, the gene, is spelled with a K because an individual named Koro was the one spelled with a K who identified this gene, but named it for a Greek goddess. So a mythological story here named Klotho, which was spelled with a C. This goddess Klotho was said to spin the web of time and thus determine kind of human lifespan. So interesting story that I always like to tell. So when we looked at this gene, we did find, in fact, that there was one genetic variant within this gene that seemed to moderate the relationship between PTSD symptom severity and accelerated methylation age. So this genotype RS9315202, if you had the less common allele or variant at this SNP, you showed an increasingly strong relationship between PTSD symptom severity and accelerated aging. But as I've mentioned before, we don't think this is specific to PTSD. So actually, when we swapped out PTSD and said, well, do we see the same relationship if we look at levels of chronic pain? Indeed, if you have increased chronic pain and you have this genetic variant, you've got this relationship between elevated levels of chronic pain and accelerated methylation age. Same thing for sleep disturbance. Relationship between sleep disturbance and accelerated methylation age is again stronger in those who have this at-risk genotype. And this finding was not even just specific to accelerated methylation age, but also held when we looked at other biomarkers of cellular aging. So again, the relationship between PTSD severity and levels of inflammation as indexed by C-reactive protein were enhanced among those with the less common allele at this locus. And we also showed that if you had PTSD severity and this less common allele at this particular locus, you showed this increased relationship between, again, PTSD severity, and in this case, loss of function or loss of structure, rather, I should say, between various white matter tracts in the brain. And these are tracts that are connecting prefrontal cortices through limbic regions. So things like amygdala and hippocampus. So here we showed loss of microstructural integrity in these white matter tracts if you have both PTSD and this at-risk genotype. So not specific to DNA methylation, but seemingly kind of organizing a cellular aging response across the central and peripheral nervous system. That was all done with looking at levels in blood. So then we actually were able to take that PTSD brain bank data that I mentioned earlier and replicate this association. So now we're looking at accelerated methylation age. We looked at dorsolateral prefrontal cortex methylation. We looked at ventral medial prefrontal cortex because those prefrontal regions are, particularly interesting to us studying PTSD and other psychiatric conditions. And then we also looked at motor cortex. And what we did was find the exact same relationship. Again, we looked at every variant again, and we could not find another one that mirrored this relationship. But this particular genetic variant, if you've got that variant, whether you have one or two copies, we show sort of a stepwise increase in risk for accelerated methylation age in the motor cortex if you also have PTSD. And then we were really fortunate in this study to be able to work with C.D. Chen and Carmela Abraham, who are Klotho experts at BU School of Medicine. And they are bench scientists. And so they were able to then run the appropriate in vitro studies where they could really look at, is this variant, is RS9315202 doing anything meaningful? And they could show that actually it did seem to be a functional variant because it was associated with alterations in clotho-transcription. So when they spliced in that variant, again, we showed alterations in clotho-transcription. And what we know about clotho is that for most people, levels of clotho are pretty stable across the lifespan till you get to old age, where some people kind of fall off a cliff and they lose their clotho expression, they lose levels of it that can be measured, and that's what seems to be associated with accelerated aging phenotypes, including Alzheimer's. So now I've been talking to you a lot about primarily DNA methylation age, but as I mentioned earlier, DNA methylation is certainly not the only epigenetic phenomena that seems to track with chronological aging. So very recently, Kurt Zhao, who is a first-year biostat student in our lab, who previously was one of our biostats analysts and is now continuing to work with us at BU School of Public Health. He's just published this paper looking at transcriptomic age. So we're not looking at DNA methylation age, we're now looking at a different algorithm of chronological aging using RNA data, so expression data in brain tissue, again, from the ventral medial prefrontal cortex, the dorsolateral prefrontal cortex, and the motor cortex. And I haven't mentioned this yet, but throughout all of our analyses, we do a lot of multiple testing correction. So the effects that I'm talking to you about today are those that have survived correction across these different brain regions, different algorithms, different genotypes, and so on. So in this study, we were able to show that if you have PTSD or depression, you have this accelerated transcriptomic age in ventral medial prefrontal cortex. And then actually we looked at what was accelerated transcriptomic age associated with in brain tissue. And we saw that in the same brain region in ventral medial prefrontal cortex, if you have accelerated aging in RNA, in gene expression, you have loss of endothelial and mural cells. And we thought that was particularly interesting because those are two cell types that help to form the blood-brain barrier. And so this is something I didn't get into in some of our other the other slides I showed you, but we've seen this effect or variations of this effect in our other papers as well. And so we are really starting to think about accelerated aging may get across the periphery into the central nervous system through its impact on blood-brain barrier integrity. Let's see here. So I've just presented a lot of results for you across a lot of different cohorts and studies. So to kind of summarize them, what we think that we are showing across these studies is that traumatic stress, broadly defined, PTSD, internalizing, externalizing, psychiatric comorbidity, is associated with accelerated cellular aging. And we've shown this both cross-sectionally and in longitudinal data and in both blood and brain tissue. But we know that DNA methylation is not the only biomarker of the aging process that is affected. We've also shown increases in inflammation, alterations in brain morphology, changes in gene expression as well. And also that we think that these factors, these relationships may be moderated by factors like demographic ones, like age. So again, critical periods of time when you may be more likely to see this relationship, and biological factors such as genotypes that may also moderate these relationships. So it's really caused us to start to think about PTSD as an environmental factor that contributes to health disparities that we're seeing at a cellular level. But really across metabolic health, oxidative stress, inflammatory, and neural integrity in terms of the adverse outcomes that we're seeing these relationships kind of replicated in. So we're starting to think about DNA methylation age as a useful biomarker for health disparities. And we think about younger veterans, like those veterans in the TRACS cohort, as having more chronic adverse health outcomes because they've got more years, a greater burden of psychiatric stress, kind of yielding a cumulative toll on cellular aging. So it really speaks to the need to kind of get in there, screen who might be at risk for accelerated aging, and change how we when we start intervening. So one of our studies, we recommended maybe instead of screening for people for metabolic dysregulation at age 40, in these veterans with psychiatric symptoms and PTSD, we should be screening them at a much earlier age, maybe starting at age 25 or 30, so we can intervene earlier and reduce the likelihood that they're going to have this accelerated aging kind of burden over the lifespan. We also think that by looking at the kind of biomarkers that are associated with accelerated aging, we can try to understand the pathophysiology and then hopefully identify, pharmacological interventions that could reverse or slow the pace of cellular aging. To date, really, the kind of strongest evidence for alterations in the pace of cellular aging have to do with caloric restriction. They primarily come from animal studies, and that is a story that nobody really wants to hear. So hopefully we can develop novel approaches for slowing or reversing the pace of cellular aging. I want to make sure that I acknowledge all of my collaborators on these projects here at the National Center for PTSD. I primarily work with Mark Miller and Mark Logue, who are 2 other PIs in our lab, but also our biostats student Kurt Zhao and our former T32 postdoc Sage Han have really contributed dramatically to this work. The TRACS cohort is led by Bill Milberg, and Regina McGlinchey is an incredibly invaluable resource. And the Traumatic Stress Brain Research Group manages the VA National PTSD Brain Bank. Again, another really invaluable resource that we've been able to take advantage of to publish this work. All of our genotyping and DNA methylation arrays are done at the VA in Little Rock at the PAL there by Angie Stone and Steve Schickman, who are wonderful collaborators. And then also I want to acknowledge our work with the Psychiatric Genomics Consortium, the PTSD work groups, particularly the Epigenetic and PTSD and Health group that I co-lead with Jen Sumner, which are really also really useful for testing that these effects we're finding are not just specific to our cohorts. Here is a slide of our funding and other disclosures. And if you want more information, I've got some of our various websites listed here. You can also always email me. I'm easy to find at the VA, but there are other Erika Wolfs. So I'm Erika with a K and Wolf like the animal, if you want to find me there. So let me escape out of here. I'm happy to take questions.
Thank you so much. That was very, very interesting. Anytime someone talks about clotho, I always think about kidney disease, and of course,
Yes.
And actually, I actually did a focus group of patients with kidney disease and asked them, like, what are their most, big concerns? And most of the patients were like patient advocates who are involved with pharmaceutical companies and research, highly educated people who have kidney disease, and their biggest concern was cognition.
Mm-hmm.
And what are we doing to address cognition? And one of our previous presenters actually talked about, the issues of frailty and weakness in patients with kidney disease. But we know that clotho levels are low, and they get lower as kidney disease progresses. And wondered if you had thought about that, looked at that at all?
Yeah, we don't really have we have some liver function panels, but we really don't have any kidney, panels on ours. But, probably like the comorbid alcohol abuse that we see in our cohort is probably going to contribute to that. We did actually measure clotho in our cohorts. We have it in blood. I haven't actually found it to be too correlated with any of our psychiatric markers yet. But it may be that at the next round, now that this cohort's now at age 65 coming back in now, that's the data we're collecting now, we'll get that again, and we may start to see some more variability in that.
Yeah, and they have clotho levels in Haines. Did you know that?
Mm-hmm. Yeah. Yep.
Yeah.
Yeah, I think it's a fascinating story, that one.
Yeah. I think Dr. Leahy had a question. Dr. Leahy, can you ask the question, or do you want me to ask it?
I guess I can ask it. You hear me okay?
Yeah.
Okay, so anyway, great talk. It's really fascinating. A few years back, there was a lot of interest in the medical community about metformin. And, it's a readily available, safe, very inexpensive agent. And, I think even for us, and even though I'm unfortunately aging, as I mentioned, I'm fortunately not getting obese, but I suspect that I do have some degree of metabolic syndrome, depending on how you want to look at it, because I think we all get that. And so the question is where have we gone, if with the metformin story? I mean, I don't hear much about it anymore.
Yeah. I mean, someone else in the screen may be able to say more, because I'm kind of very focused on the accelerated aging story about it. And there actually have been at least one study out there looking at DNA methylation agent metformin and really showing no relationship. I would say in general, and there's also studies about anti-inflammatories and accelerated aging. The results have been very inconsistent across studies that have looked at this and across studies even within a study that have looked at cross-sectional versus longitudinal associations, and they don't replicate the cross-sectional associations they observe in the longitudinal data. So at least with respect to accelerated aging, I'd say the jury is still out, but there's no evidence yet that that could reverse, this methylation, accelerated methylation age. On the diabetes side of things, I'm less knowledgeable because I'm not specifically focused on that.
Yeah, and I guess another question would be on fasting.
Mm-hmm.
Is there a lot of,
Fasting, like
does
intermittent
fasting
fasting.
affect methylation?
No, methylation isn't change that dynamically, so it's not like, so like our samples that we collect, our fasting samples, because we're also getting all the metabolic profiles at the same time. So, gene transcription is much more dynamic, but methylation changes, with age, but changes more slowly. So it's not going to impact our levels of methylation. Those animal studies that I mentioned on caloric restriction are, over the period of weeks, a significant proportion of that rodent's lifespan.
Okay. Okay. Does anybody else have any questions?
Yes. Hi. Thank you for such a wonderful talk. I just was interested in one of the findings you were talking about with clotho and methylation in the motor cortex.
Mhm.
I've recently been interested in the motor cortex as I have a TMS study that's delivering TMS to the motor cortex for mild traumatic brain injury and chronic pain. So I was just kind of wondering how the motor cortex got brought in with some of the other outcomes and things that you've been looking at.
Yeah, well, as things sometimes work out, the motor cortex was brought in initially as a control brain region, right? And so we were more focused on ventral medial and prefrontal cortices. However, for something like clotho, where there are definitely motor kind of phenotypes associated with loss of clotho, it became relevant for us to look at. So when we wound up, we didn't see this effect in methylation in ventral medial or dorsolateral prefrontal cortex. We controlled in our analyses. We corrected the p-value across the three different hemispheres that we were, not hemispheres, three different regions that we were looking at. And that's how we got onto this story. So whether or not that is an effect that we saw specifically in motor cortex, because we were looking at clotho as a moderator, is up for debate. We really could use another brain bank cohort to look at that, but of course a PTSD brain bank cohort, they're a little few and far between.
Okay. Dr. Gilmer, do you have a question?
Yes, I did. Thank Dr. Wolf. This was absolutely phenomenal. Thank you. I'm a psychiatrist and mood disorder specialist, and have long been interested in cognitive impairment in chronic recurrent affective disorders. Did some work in euthymic bipolars going back 20 years. And, one of the things, when I came to the VA recently, it struck me how, few veterans have just one behavioral health diagnosis. I mean, they have one, they have 4 or 5. And when you were commenting on, you noted that the hyperarousal seemed to be driving some of the accelerated methylation. I thought of like the Gauge BD groups recently. I think Lisa Eilers was the first author on a paper that showed mixicity was strongly predictive. And certainly with chronic recurrent affective disorders, the more recurrent they are, the more arousal and activation. And Kathleen Marikangas has been doing some work looking at sort of transdiagnostic arousal activation at the intramural program. And I'm conjoining 2 questions here, but I'm wondering if we're dealing with a highly co-occurring or comorbid or transdiagnostic population, what approach would you recommend as far as research for capturing that, if it is some common phenomenon? And I was especially struck by your recent paper that you were co-author on about the high tops and the RDocs. And so I wondered if you discussed the dimensionality of that, and if
Sure.
there may be some other ways for us in these messy veteran populations to approach this.
Yeah, so I didn't talk about, that line of our work here at all. But the other part of the other hat I wear when it's not accelerated aging is this kind of thinking about the structure of psychopathology and using multivariate data analyses like latent variable models to try to do a better job of modeling those phenotypes. Because one thing that we know is that these discrete DSM, yes, no, diagnoses are really far away from any biomarkers that we're interested in. And they are, of course, developed in a boardroom. And so we know they're not what the genotypes are coding for. So we want to try to do a better job of modeling that. So that paper that you're referencing, led again by Sage Han, was that's in Clinical Psychology Review, I think, is one where we tried to say, and we've done this to various degrees, we think we need to do a better job of reflecting the broad dimensionality in psychopathology and the relationships between PTSD and a number of different psychiatric conditions. So you can do that in measurement models or confirmatory factor analyses where you can model, internalizing disorders. You have to measure them all. You've got to, give a skid in addition to a cap so that you've got all of them. And then you can identify people's sort of scores on these latent variables, which reflect the common elements across, say, depression and anxiety and other mood disorders versus substance use and antisociality. You can measure where an individual lies on that spectrum. And then in the paper that you're referencing, we also kind of have made the argument that, when we study these things, we usually start at like no PTSD and go up to like high PTSD. And that seems to be a problem because factors like resilience are actually very strongly correlated with PTSD, negatively correlated. And so in another paper that we did, I used to do some twin modeling. I don't do too much of it anymore, but we worked with the Vietnam era twin registry, and they have a measure of resilience in there, and they have their measure of PTSD. And we were actually able to show that the genetic factors that contribute to PTSD 100% overlap the genetic factors that contribute to resilience following trauma exposure, meaning that we think the dimension really should be, resilience, adaptive functioning on one end, and high symptom severity on the other end. Because if we're not modeling that bipolar dimension, we're going to truncate the range of our symptoms. And so when you go to do, let's say, a genetic association study, like a GWAS, a genome-wide association study, and you're just looking at, PTSD, no PTSD, or no symptoms and severe symptoms, you actually only have your variance on this half of the dimension, and you're missing the other side of it. And that statistically will reduce your statistical power to identify genetic and any other correlates of it. So to answer your question more directly, we do assess everything. It takes a long time, but we assess across, common comorbid conditions using SCID. On a few studies, I've tried to shorten the study by giving like an MMPI instead of doing the SCID, or if I don't have enough interviewers, because that's got these internalizing, externalizing, and thought disorder dimensions that are the higher order dimensions on the measure, so that I can still try to model the psychiatric comorbidity and think about the extent to which, symptom severity is really contributing to this increased overall expression of different types of conditions.
Thank you.
Hope that answers your question. Thanks.
Dr. Patel, did you have a question?
Oh, yes, I did. I'm not sure if I missed this, but did you touch on anything that can decelerate aging?
Yeah, it's much harder. I haven't done that work myself, but other people have worked on it. So really the only thing that I would like put my kind of money down on from the literature thus far is in the animal literature focused on caloric restriction. So that is not, as I said earlier, like that's not a story anybody really wants to hear. There doesn't seem to be many effects for exercise. There are some weak effects for nutrition, like specific types of nutrition as opposed to overall caloric restriction. But there is a group, Steve Horvath left his academic position, is at Altos Labs now, and that's their mission is, reprogramming the epigenome to slow down the aging. I don't do that stuff. I try to tag on to people who are doing treatment research. One of the things I think is important to note, it's really hard to do treatment research to see how accelerated methylation age could be changing is you have to have a long enough follow-up period. We would not expect your methylation age. You do a 12-week intervention, you're not going to see a difference in your methylation age in 12 weeks. We don't even have enough of a sensitive measurement to be able to say this time point is different from this time point in terms of your estimate of methylation age. So you need to keep people in your studies, 2-5 years or so on. And most treatment studies are not designed to do that. That becomes challenging.
Thank
Jury's
you.
still out. Yeah, you're welcome.
Interesting. All right, any other questions?
There's a question from Dr. Adams in the chat.
Oh, she said, we have long thought of aging with immunosenescence, reduced immune response. In some older work stress reduces some aspects of immunity. Intrigued by the concept of enhanced or dysregulated immune function with age. How well is this understood?
Yeah, actually the immunosenescence piece of aging, definitely a pretty big part of the story, well researched. It's connected strongly to the kind of inflammaging because we know that with increased, that there's this bi-directional relationship, right? So as cells start to die off or become immunosenescent, they secrete an inflammatory kind of profile. And then you increase the inflammation, that increases oxidative stress, and you have more immunosenescence. One thing that is kind of interesting, there's a paper out there by Steve Horvath, again, where he looked at, is accelerated methylation age, is it an immunosenescent kind of process, or is it really like, is it pathology, or is it really like a real acceleration of aging? So the same process that you would see in aging, but happening faster. And he compared DNA methylation age to shortened telomere length. And what they showed, in a bench science kind of study was that the, shortening of the telomeres seemed to be kind of an immunosenescent kind of process, whereas the acceleration of aging appeared to be more about not so much a pathological process, but a sped up process. So a normal process that's been sped up. So two different things. However, the immunosenescence, all of these factors kind of, we know, correlate with aging. So I think they're tapping different elements of kind of biological aging. So they are occurring at around the same time points, but are driven probably by different, kind of biological factors.
Okay. All right. I think we're gonna go ahead and stop there because we're at our time, but I know that you're gonna stay on to talk with
Sure.
some of the people in Bill Gilmer's group. So thank you everyone for coming, and if you were planning on meeting with Dr. Wolf after her talk, please stay on this
Yes,
Zoom
stay on.
call.
Right. And Dr. Wolf's been very generous with her time. So we'll make it efficient for the small group who can stick around. We have a monthly behavioral neuroscience research group that gets together, and when we have external speakers in particular. And so this seemed like a wonderful opportunity to for some of our folks to stick around and have a chance to chat with you.
Yeah, that's great.
So thank you. And we are not recording this. Is this correct, Holly?
Oh, yeah, let me stop.
Yeah, so we'll everything can be off the recorded record.